Happy Friday. Hope you had a good Thanksgiving break. Does anybody remember Thanksgiving? Is everybody ready to get this over with? Yes. Get some enthusiasm for that, right? So I have promised to do overview sort of weekly sessions, but it occurred to me since it is the end of the term that maybe it would make more sense for the one this week to be at the end of the week instead of doing it in the middle and then doing another one. So I'm inclined, I haven't set a time yet, but I'm inclined to either do it Friday evening or Saturday as an overview, like I've done with the previous materials. And then I'll have a review session probably, I'm thinking Sunday. And I'll videotape, of course, all of these. But does that seem reasonable to you? Any thoughts, comments, concerns? Okay, so I will try to get a room set up to do that, and we will go from there. So we are in the home stretch. We have 3 lectures finishing up carbohydrate metabolism. We will get 2 pathways done in that time. And I think you'll see that the first one looks a little bit more complicated than it actually is. The second one you'll see is not complicated at all. It's probably the simplest pathway that we talk about, although the regulation of it is a little bit complicated. Okay, so without further ado with that, let us dive into the material. It's not going to be a bad week in terms of total content that we will cover. By the way, some of you have asked about percentage of the exam that will be old and new material. I think I said this before, but I'll say it again. And that is that the new material since the last exam will be roughly the percentage of the coverage of that in the entire term. So that's roughly 25 to 30% of the term will be new material of the exam will be new material, and the remainder of the exam will be material that we've already had. So that's the strategy for the exam. And as I noted, you will have a note card to use on the exam, and you must get the note card from me. And no, I didn't bring the note cards today. But you must get the note card from me. That's important, okay? So make sure you get and use a note card from me. I will tell you beforehand that they're big note cards. They're 5x8s, so if you want to start prepping one that you can copy onto the note card that I give you, that's the size of the note card that you'll be working with. So it's a pretty good size note card. Okay. Well, biochemistry of carbohydrates. So far we have talked about glycolysis, we've talked about gluconeogenesis, and we have talked about the regulation of those. And with the regulation of those, you start to see how your body is responding to need for energy. Making glucose when there's abundant energy, and using glucose when it needs ATP. So those are the 2 guiding principles that lead us through all of carbohydrate metabolism. We'll see that it leads us through essentially all energy metabolism too. We do the same things with fatty acids as we do with carbohydrates. The strategy is a little bit different, as we will see, but the basic idea is the same. Burning fatty acids when we need energy, making fatty acids when we have energy. Okay. Well, I want to start talking about a pathway that is called the pentose phosphate pathway. I abbreviate it PPP, and you can abbreviate it PPP as well. The pentose phosphate pathway is a pathway that I think is largely misunderstood, and I do try to keep it simple for you, and I promise I will keep it simple for you. There are actually 10 different structures that appear in the pentose phosphate pathway, and I don't expect that you're going to memorize those structures. Okay? So that's a given. You're not going to memorize those structures, although if I asked you about glucose 6-phosphate, I would expect you would know that one because you've already known glucose's structure, and putting those phosphate on position 6, just like I talked about in glycolysis, is not an unreasonable thing to do. The other structures I'm not as concerned that you memorize because I don't think they serve any purpose for memorizing them. As we look at these structures, we see some common things. First of all, you'll notice that 9 of the 10 structures all have a name that ends in OSE, or have a part of their name that has OSE in it. All right? That OSE, you remember, refers to a sugar, And so it means that 9 of the 10 components of this are sugars. The one that's not a sugar is sort of an intermediate, as we shall see. And technically, it is a sugar as well, although its name wouldn't reflect. It's a modified sugar. Its name wouldn't actually relate to that. Okay. Now, that means that this is a pathway that largely involves sugars, sugar phosphates, or modified sugars. That's what the pentose phosphate pathway is all about. All right? As I go through and I show you the pathway, I am not going to expect you to memorize the steps of the pathway. I'm not going to expect you to redraw the pathway. You'll see the pathway is kind of convoluted. The pathway is more of an intersection of things than it is a pathway. I'll emphasize that in the last part of what I have to say about it. It's more of an intersection of other pathways than it is a pathway all by itself. We call it a pathway, and I hope you remember from what I told you earlier that pathways are inventions of man. Okay? We can look at this and say, okay, this connects to this, connects to this, connects to this. This is a pathway. We could do the same thing with Albany is over in that county, Corvallis is over in this county, right? So county lines are things that are drawn by men. I should say men, people, okay? County lines are things that are drawn by people, right? And pathways are as well. So delineating something as a pathway is a completely artificial thing. So when I think about the pentose phosphate pathway, I think about it in terms of being something that is a connection, an interchange. And you'll see that this is one of the biggest interchanges of cells. Okay? Just like where we could have several major freeways converging and things going out a variety of directions from those freeways, so too can we see the pentose phosphate pathway being an interchange of many different metabolic pathways. Now, what the pentose phosphate pathway does is it allows for rearrangement and exchange depending upon an organism's needs. It does things depending upon an organism's needs. And that's the first pathway we've seen in this regard. This pathway doesn't have a single direction. It doesn't have a single direction. Gluconeogenesis made glucose. Glycolysis broke down glucose. When we do glycogen metabolism, we'll see that glycogen synthesis makes glycogen, and glycogen breaks down glucose, right? The pentose phosphate pathway has several different directions it can go, and the direction it actually goes depends upon cellular needs and availability of materials. Now, the pathway, as I will talk about, has several important features associated with it. Number one, it is the cell's most important source of NADPH. NADPH is related to NADH. It has a phosphorus on it, whereas NADH doesn't have that. And you remember that NADH is a reduced form of NAD, meaning it's carrying electrons, carrying a proton. And so NADPH is a reduced form of a molecule we call NADP. And so why do we have NADPH and NADH? The answer to that question is that cells tend to use NADPH for biosynthetic reactions. It uses it for biosynthetic reactions. We'll see it's commonly used in making fatty acids next term. So cells must therefore have a way of making NADPH, and the pentose phosphate pathway is the primary pathway for doing that. A second thing that the pathway is important for is for making precursors for nucleotide metabolism. This pathway is the cell's only synthetic source of making ribose 5-phosphate. Ribose 5-phosphate. Ribose 5-phosphate is needed. It provides the ribose for making nucleotides. So this pathway is very important for providing that material. A third thing that this pathway provides is it actually provides an intermediate necessary for synthesis of aromatic amino acids. Provides an intermediate necessary for synthesis of aromatic amino acids. And then last, sort of parenthetically, this is the fourth one coming up. Parenthetically, this pathway is important for swapping sugar configurations. We'll see how 5 carbon sugars can be made into 7 carbon sugars, how 6 carbon sugars can be made into 4 carbon sugars, and a lot of possibilities with that. And again, the direction that the pathway goes is a function of the cellular need and availability. I can't hammer that point home hard enough to you. So I say all that before I actually show you the pathway. Here's the pathway. Now, books write this pathway in a variety of ways, and I've actually spent some time thinking about this pathway, and this is my own version of the pathway. As I noted, it doesn't have a single direction. It has some complexity to it, and I'm going to simplify that for you in just a minute. But as I said, you're not going to regenerate this pathway for me, okay? It wouldn't serve any useful purpose. But I think you should have, in learning about the pentose phosphate pathway, a broad perspective, a broad overview of what the pathway looks like and what the pathway does. Okay? Now you'll notice in red on the screen there are 4 different pathways connected to it. There's actually 5 things, but there's 4 different things, 3 of which are unique. Okay, so the unique ones are nucleotide synthesis, glycolysis, gluconeogenesis, and tryptophan synthesis, which includes the aromatic amino acid synthesis. Okay, those 3 pathways use intermediates and provide intermediates to this pathway. So these are multi-directional. Not only do the things exit and go to the individual pathways, but individual pathways can provide those materials to feed it. Okay? So we could imagine, for example, that if the cell had a lot of intermediates from glycolysis and gluconeogenesis, that those intermediates could feed the pathway and go and make ribose 5-phosphate for nucleotide synthesis. And I'll show you how that happens later today. Okay? The numbers on the screen indicate enzyme numbers, and that doesn't really matter for our purposes. You can see that there are 7 different enzymes that are used in the pathway. So let me break it down, this complex pathway, break it down into smaller components, and then talk about those components, because I think there is where the information of the pathway comes about. Okay? The first part of the pathway, if we think about the top being the beginning, and there's not really a beginning or an end to this pathway, but the first part I want to talk about is the part called oxidation. Okay? The pathway only has 2 reactions involved in oxidation, and you can see that in those 2 reactions of oxidation, that NADPH is produced. That's no surprise, because oxidation is going to cause something to be reduced, and the something that's reduced is NADP producing NADPH. Okay? The second part of the pathway that I want to mention is what I call isomerization. Okay? Isomerization involves rearrangement of molecules. It doesn't involve swapping anything. It simply involves rearranging. We saw, for example, in glycolysis, that glucose 6-phosphate was rearranged into fructose 6-phosphate. And the types of rearrangements that I'm talking about here are very much like the rearrangements that we have in glycolysis and other pathways. We're simply taking a molecule and changing its configuration. Nothing complicated about that. The third part about the pathway that I want to mention is what's called carbon rearrangement. And this involves swapping of carbons between individual molecules. And I'll show you some reactions for that. Again, I'm not expecting you to memorize structures with any of these. Okay? But the enzymes that are used in the carbon rearrangement part of the pathway are useful for other purposes, as we shall see as well. Okay? And these are some very interesting enzymes. Okay. Let's talk about the oxidation part of the pathway. In the oxidation part of the pathway, we're producing NADPH. And there are actually 3 reactions, 2 of which are oxidative in nature. The first one starts with our old friend, glucose 6-phosphate. And glucose 6-phosphate, okay, is linked to glycolysis, right? Linked to glycolysis. We could take this out of the glycolysis pathway and use it here. Alternatively, if we had excess glucose 6-phosphate here, we could put it back into glycolysis, so it could either be broken down or made into glucose. Okay? This oxidation produces an intermediate known as 6-phosphogluconate gamma-lactone, which you don't need to know the name of. All right? And that intermediate is converted into 6-phosphogluconate, all right, which is shown here. There's an enzyme that does that, but the enzyme isn't really relevant, so I didn't put it on there. But in the next oxidation, 6-phosphogluconate is converted into ribulose 5-phosphate. There's your second oxidation. Okay. Glucose 6-phosphate dehydrogenase is a very important enzyme, and I expect that you will know the name of that enzyme. Okay. That enzyme is another way of handling glucose 6-phosphate. Okay? Yes? Okay, why the CO2 on the second? You're one step ahead of me. All right, why the CO2 on the second reaction? This is the only decarboxylation that occurs in the pentose phosphate pathway, because what's happening is you're converting a 6-carbon sugar into a 5-carbon sugar. Okay? That oxidation reaction produced the equivalent of a carboxyl group in that first oxidation. It was a gamma-lactone, so it's not a carboxyl, but it's the equivalent of a carboxyl group on that first oxidation, which means that the next oxidation is readily able to happen, and the process releases a carbon dioxide. So we've gone from a 6-carbon sugar to a 5-carbon sugar. Everything else that happens in the pentose phosphate pathway works with those carbons. Everything. Okay? So we lose 1 carbon if we start at the top and we go all the way through it. As you will see, we can run this pathway without even going through this part of it. Because again, the direction of the pathway doesn't have a beginning or an end. It depends on what's available, and I'll show you that as we get going along. In the next part of the pathway, we see rearrangements happening. Rearrangements. Here's one of the rearrangements. Ribulose 5-phosphate is converted into ribose 5-phosphate. And ribose 5-phosphate, I told you, was important for nucleotide metabolism. Okay? Isomerization is what's happening here. Again, don't worry about the structures, but I just show them to you. There's ribulose 5-phosphate, and there is ribose 5-phosphate. What's the reaction that you've seen that mirrors that one? What? Fructose 6-phosphate and glucose 6-phosphate, right? So you're seeing, in this case, a ketone being converted into an aldehyde. In the case of glycolysis, we saw the aldehyde converted into the ketone. Or if we looked at gluconeogenesis, we saw the ketone converted into the aldehyde. Okay? So this is simply a keto-aldo rearrangement, just like we saw in glycolysis. There's the enzyme. I'm not too concerned about the enzyme name. So, as I said, this reaction is very important for nucleotide metabolism, okay? Because this is the cell's source of ribose 5-phosphate. Here's another rearrangement. Ribulose 5-phosphate can also go to xylulose 5-phosphate instead of going to ribose 5-phosphate. Provides an alternate pentose source. And that means that if this organism, for example, gets in its diet something that has a bunch of xylulose, it can convert it to ribulose 5-phosphate, and ribulose 5-phosphate can be converted into ribose 5-phosphate. So again, you see the flexibility of the pathway. So this is a major interchange for all kinds of things coming in and going out. And again, I'm not worried about the enzyme name. I've also shown in this figure, as I did on the last one, I think, at the very top, the overview of the pathway and where these individual reactions play in, and I've marked them in yellow, as you can see. Now, the next part of the pathway, I think, is actually the most interesting part of the pathway, and this is what I call the carbon rearrangement part of the pathway. Carbon rearrangement part of the pathway. Now, from the names, you don't really see what's happening here. Ribose 5-phosphate plus xylulose 5-phosphate gives cetoheptulose 7-phosphate plus glyceraldehyde 3-phosphate. But when we look at the structures, we start to see why this is kind of an interesting thing. Ribose 5-phosphate looks like this. If you count the carbons, you see it has 5 carbons. Xylulose 5-phosphate also has 5 carbons. And you remember that, of course, it was produced by a rearrangement of a 5-carbon molecule. So we have 2 molecules to start with here, each of which have 5 carbons. In the reaction, a 7-carbon molecule is produced called cetoheptulose 7-phosphate, okay? And a 3-carbon molecule is produced called glyceraldehyde 3-phosphate. Notice we started with 5 and 5, and we ended with 7 and 3. That means that some rearrangement, movement of carbons occurred from one molecule to another. If we take 2 off of one of the 5s, we end up with a 7 and a 3. And that's exactly what happens here. The enzyme catalyzing this reaction is a very important one. It's called transketolase. It's an enzyme that I think you should know the name of. Okay? Transketolase catalyzes the transfer of keto groups. And these keto groups are 2-carbon pieces. Okay? The enzyme requires a coenzyme called thiamine pyrophosphate, TPP. And we'll see this cofactor or this coenzyme come up in other reactions next term. Carrying of 2-carbon pieces is typically done by thiamine pyrophosphate. And yes, thiamine is a vitamin. Okay? Last time I talked about something that carries 1-carbon pieces, and that was biotin. Biotin was carrying carboxyl groups. So here's something now that's carrying 2-carbon pieces. It's thiamine pyrophosphate, and it's a coenzyme for the enzyme transketolase. Okay? This enzyme also occurs in the Calvin cycle. The Calvin cycle is what's known as the dark cycle of photosynthesis, and it's in this cycle where glucose is actually made. Now, we don't talk about photosynthesis in this class, unfortunately, but I will tell you that several reactions of the Calvin cycle overlap with reactions of the pentose phosphate pathway. So if you learn the pentose phosphate pathway, you've gone a long ways towards understanding the Calvin cycle. Now, let's take a look and see what's actually happening with these molecules. We start at the top with ribose 5-phosphate. Come on. Oop, I hit the wrong thing. No wonder. All right. Here's the part that's going to move. All right? You see it's a keto group, and you see what's left over after it moves. So now if we follow the pieces, what happens? We see the keto group gets put onto the 5-carbon piece from ribose 5-phosphate, and we're left behind with a 3-carbon piece. Notice that the arrows go both directions, meaning that we could easily start with cetoheptulose 7-phosphate and glyceraldehyde 3-phosphate and go backwards to make ribose 5-phosphate and xylulose 5-phosphate. But transketolase is the key to this reaction because it's the carrier of the 2-carbon piece from one molecule to another. Okay? Here's another interesting reaction. Okay? In this reaction, we start with one of the products, or the 2 products of the last reaction, cetoheptulose 7-phosphate and glyceraldehyde 3-phosphate. You remember those were 7 carbons and 3 carbons each. In this reaction, the products have 6 carbons, the fructose 6-phosphate, and 4 carbons, the erythrose 4-phosphate. Well, you can do the math and figure out that what's moving here are not 2 carbons, but 1 carbon. The 1 carbon being moved, we will see, is moved by an enzyme. There's the 7, there's the 3, called transaldolase. Transaldolase is another important enzyme to know the name of. In this case, it's carrying an aldehyde group. Okay? It's carrying... Well, actually, that's a little misleading. It carries something that becomes an aldehyde group. So we'll see it's not an aldehyde, but it does become an aldehyde group. The products have a 6-carbon and have 4 carbons. In order to produce this 6-carbon molecule, here's what happens. Okay? Don't worry about the thing at the bottom. Okay? Just for your information. There's the 1-carbon piece. That will become why am I saying this wrong? That will become an aldehyde. Okay? There we go. So we see that CH2OH on the top is transferred down to the molecule on the bottom and becomes an aldehyde. That's the aldo part of the transaldolase. And we see what's left from that is a 6-carbon piece known as fructose 6-phosphate. And the 3-carbon piece that gets joined to the 1-carbon piece is glyceraldehyde 3-phosphate. So we've moved 1 carbon from cetoheptulose 7-phosphate onto glyceraldehyde 3-phosphate to make erythrose 4-phosphate, and we're left behind with fructose 6-phosphate. Now, these reactions are also reversible. Okay? So if the cell, for example, had a bunch of fructose 6-phosphate from glycolysis, for example, and it had some erythrose 4-phosphate, that reaction could go be used to make cetoheptulose 7-phosphate, glyceraldehyde 3-phosphate, which could go backwards in the previous reaction. So we see flexibility of these reactions going back and forth. Now, what I want to do in the last part of what I'll say about the pathway is show you how this pathway really is incredibly flexible in terms of meeting cellular needs. Okay? Let's imagine we have a situation where the cell has high levels of NADP+. Okay? When that happens, and the cell needs to do some nucleotide synthesis, it can't rely on glucose 6-phosphate for producing the ribose 5-phosphate necessary for nucleotide synthesis. Well, what are the alternatives available to the cell? Well, the cell may be readily going through glycolysis, gluconeogenesis, and has a variety of intermediates available there. Those might be fructose 6-phosphate and glyceraldehyde 3-phosphate. Okay? If these two fed into the pentose phosphate pathway, I'm going to show you how those could be used to make ribose 5-phosphate necessary for nucleotide synthesis. First, we would see the glyceraldehyde 3-phosphate and the fructose 6-phosphate combine, as you see here, to make the xylulose 5-phosphate and erythrose 4-phosphate. That's another reaction involving transaldolase. Haven't shown you that one, but it's another one. Okay? Erythrose 4-phosphate can combine with fructose 6-phosphate, because we've just made erythrose 4-phosphate, to make cetoheptulose 7-phosphate and glyceraldehyde 3-phosphate. These guys go backwards to here, and look, we've made an abundance of ribose 5-phosphate. So even though part of this pathway was blocked because we didn't have enough material to go forward, okay, this pathway would lead to the glycolysis gluconeogenesis intermediates would lead to the production of ribose 5-phosphate. So we see how things can come in from different directions and feed pathways as necessary. Okay? Let's imagine we have nucleotide breakdown going on in the cell. The cell has just encountered another cell that it has engulfed. And that other cell was full of DNA and RNA. Well, our cells are full of nucleases to break things down. And when they break things down, they get a lot of nucleotides sitting around doing nothing. Cells can break those nucleotides down into ribose 5-phosphate. And if they break it down into ribose 5-phosphate, what's going to happen? Well, there's the breakdown right there. Okay? We can see ribose 5-phosphate can be isomerized partly into ribulose 5-phosphate, which can be made partly into xylulose 5-phosphate, And now we've got both ribose 5-phosphate and xylulose 5-phosphate. You can probably figure out what's going to happen. These two guys go here. These two guys go here. And we've made erythrose 4-phosphate for aromatic amino acid synthesis. So in this case, we've had nucleotides that have contributed intermediates necessary to make molecules like tryptophan and phenylalanine. A third scenario. Imagine we have very little G6P. The cell is trying to make glucose, has very little G6P. But it's also got a bunch of nucleotides that's broken down. What can it do? Well, it makes a lot of ribose 5-phosphate. Like before, some of that can be converted into ribulose 5-phosphate. Okay? And by the way, that reaction, as I noted, is not readily reversible, so we can't just go upwards. All right? Ribulose 5-phosphate goes into xylulose 5-phosphate. We have the same thing. We have the same thing. And where does fructose 6-phosphate go? It goes to gluconeogenesis. So we see now that the products of nucleotide metabolism can be used to make glucose or give intermediates to break down in glycolysis, just like those intermediates could be used to make nucleotides. So when I talk about this being an interchange, an exchange of pathways, this is really what I'm talking about. It doesn't have a beginning. It doesn't have an end. It's an interchange of individual molecules and pathways. Okay. Questions about that? Yeah. I'm sorry? Oh, good question. Are the delta G for all these reactions relatively close to zero? It certainly is for the non-oxidation reactions. The other reactions is going to produce a bit of energy, and so they will have a little bit more consideration. Yep. But yes, these reactions readily occur with a delta G 0 prime fairly close to zero. Okay. Other questions? Okay. Well, let's see, how are we in time? That gives me some time to start talking about glycogen metabolism. So that's what is going to be our last focus. And glycogen metabolism, as I said, is one of the easiest pathways to learn. One of the easiest pathways to learn. Okay? You've seen a little bit about glycogen already. Glycogen is the animal equivalent of amylose and amylopectin. Primarily amylopectin, because amylopectin is the plant polymer of glucose that has branches. You recall that amylose, for example, was a polymer of glucose with only alpha-1,4 linkages. Amylopectin had glucose not only in alpha-1,4 linkages, but about every 50 residues or so, it had a 1,6 branch. And at the time, I told you that glycogen was related to it because it has many, many alpha-1,6 branches. And it turns out for animals that's really important because, as we will see, the breakdown of glycogen starts at the ends and moves inwards. The breakdown of glycogen starts at the ends and moves inwards. Okay? In order for that to happen, I shouldn't say in order for that to happen, a byproduct of that is that if you start working at many ends, you can release many more glucoses simultaneously than if you only have one end. Now, this fits in really well to the diverse energy needs of organisms. An organism that needs to escape that grizzly bear that we sang about last time, okay, is going to need a lot of glucose very quickly. A plant that is sitting around might want to escape the grizzly bear, but it can't run. It doesn't have that immediate need for glucose like the animal does. So having a very branched glycogen makes a lot of sense because that organism can dump a ton of glucose into the bloodstream very quickly. So branching is very important for glycogen's structure. Very important consideration of glycogen structure. What you see on the screen is a depiction of glycogen's overall structure. At the very center of the molecule is a protein called glycogenin. Glycogenin. Okay? Glycogen is stored, as I've noted before, in the liver and muscles primarily. Because it's the muscles, for example, that need that immediate energy. So if the glycogen is inside of them, breaking it down gives them glucose immediately. Okay? And the liver is important to have glycogen because the liver has to balance the glucose levels for the entire rest of the body. So because it has that need to provide glucose, the liver has a lot of glycogen contained within it. As I said, it's similar to amylopectin of plants, and in the liver helps to modulate blood glucose levels and respond to needs. Now, there's that glycogenin protein that I talked about. It turns out we can't just start synthesis of glycogen using only glucose. It has to be attached to this glycogenin protein in the very first step. I don't talk about it anywhere except right here. But just remember that when we start glycogen synthesis, glycogenin is what does the starting. You can see that the structure of glycogen is fairly complex. There can be up to 20,000 glucose units with a lot of branching. And if you look at the number of ends that are sticking out there, and that's only a small number of glucoses, you begin to see, oh, wow, yeah, glycogen has a lot of ends, and so when these enzymes start working on ends, it's really going to chew this back and release a lot of glucose very quickly. Now, I lied just a little bit. Breakdown of glycogen doesn't release glucose. It releases something that can be made into glucose. The very first step in the glycogen breakdown pathway involves an enzyme called glycogen phosphorylase. Glycogen phosphorylase catalyzes the reaction that you can see on the screen. And yes, this is a very important enzyme. All of the enzymes of glycogen metabolism are important. Okay? So you should know the names of the glycogen metabolizing enzymes. This is a very important one. We'll spend a fair amount of time in the next lecture talking about this enzyme. This enzyme catalyzes an interesting reaction. It catalyzes a reaction that you haven't seen before. It's called a phosphorylasis. A phosphorylasis is like a hydrolysis, except for instead of using water to break bonds, a phosphorylasis uses phosphate to break bonds. And in the process, the phosphate gets attached to one of the molecules, just like in a hydrolysis, water gets attached to one of the molecules. In this case, the phosphate gets attached to a glucose and creates a molecule called glucose 1-phosphate. That glucose 1-phosphate, as we'll see in the next reaction, can readily be converted into glucose 6-phosphate. And glucose 6-phosphate, you know, is only one step away from glucose. Okay? Here's that reaction. This is the reaction that converts glucose 1-phosphate into glucose 6-phosphate. It's catalyzed by an enzyme called phosphoglucomutase. What does the last part of the name of the enzyme tell you about the intermediate here? What's the intermediate of this reaction? What's that? The intermediate molecule of this reaction. No, it's not 2,3-BPG. But you're thinking in the right direction. Who wants the extra credit? Yeah. G16P. Yeah. Glucose 1,6-bisphosphate. Because a mutase, remember, puts a phosphate on and then takes the other one off. So the intermediate here, and there's an extra point of credit right there, okay? The intermediate here is glucose 1,6-bisphosphate. And yes, that can be released freely, just like 2,3-bisphosphoglycerate could be released freely. Okay. All right. Glycogen breakdown is what I'm going to finish with glycogen breakdown, and then I've got something for you, okay? The last step of glycogen breakdown is a little unusual. Glycogen phosphorylase has limits in terms of how close to a branch it can actually cleave off glucoses. Remember that branches have alpha 1,6 bonds, and all the other bonds in the molecule are alpha 1,4. Only the branch itself is 1,6. You can see it's marked on the screen, the alpha 1,6 branch. Okay? So let's imagine we started with a molecule at the very top, and glycogen phosphorylase started working on it. What you would see is that the molecules shown in blue would readily be cleaved off one by one. Each one of those would become a glucose 1-phosphate. I happen to have 10 of them there. You add 10 phosphates, you get 10 glucose 1-phosphates. The product of that reaction leaves you the molecule shown right below it. You can see that it has a long chain of purples on the bottom, and above it, it has 4 glucoses, 3 labeled in yellow and 1 labeled in red. Okay? That is as close to a branch as glycogen phosphorylase will cleave glucoses. That's as close to the branch as glycogen phosphorylase will get. If cells didn't have another enzyme to go further to the branch, then a lot of the sugars in glycogen would not be of any use at all. Okay? Well, fortunately, the cell does have an enzyme that will handle that. And it's got a mouthful of a name, but I call it debranching enzyme, and you can too. What does debranching enzyme catalyze? Okay? Well, there's the glycogen phosphorylase. The debranching enzyme starts with that material at the top, as you can see. It clips off the 3-carbon piece and actually moves it to the other part of the glycogen molecule. There's the move. Okay? That leaves behind a single glucose molecule on the top, shown in red. Okay? That single molecule of glucose shown in red is clipped by the debranching enzyme to release a free glucose. Now let's think about what's just happened. We had, glycogen phosphorylase used phosphate to break bonds between individual glucoses, but this guy is using water. That has some significance. First of all, let's think about the phosphorylasis reaction. Why do you suppose cells use phosphate instead of water to break off those first glucoses? Is there an advantage to the cell to do that? Yes, Leah. So she says there's an energy consideration, and that is correct. But there's actually more to the story than that. If I clip off a glucose using water, I'm going to end up with glucose, right? If I clip off a glucose using a phosphate, I end up with glucose 1-phosphate, which becomes glucose 6-phosphate. Does that have higher energy than glucose or lower? Higher, right? I've magically put a phosphate onto glucose without using ATP, folks. Remember we went from glucose to glucose 6-phosphate? I had to use an ATP with hexokinase to make that happen because it took energy to make that happen. Why doesn't it take energy to make this happen? Any thoughts? Leah. Well, it is catabolism which is releasing energy, but what's releasing energy here? Breaking of what bond? Okay, there's an extra credit point. So it's the alpha 1,4 bond has energy associated with it. And the energy of breaking that bond is used to put the phosphate on. So the cell is being very, very efficient. Very, very efficient. So the next question then is, well, why doesn't the cell do the same thing with the last glucose with debranching enzyme? Why doesn't it do it there? It's a different bond and has less energy than an alpha 1,4. There's another extra point credit, okay? It has less energy. So we can see right here on the screen that an alpha 1,4 bond has more energy in it than an alpha 1,6 bond does. That's pretty cool, right? So cells use everything available to them, and they don't waste energy. There's not enough energy in an alpha 1,6 to readily produce a glucose 1-phosphate. So it uses water because it has to. Okay? Pretty cool. That's the upshot of the breakdown of glycogen. Okay? You notice that what's left at the end of that is a molecule that now has a lot of alpha-1,4 bonds that can be broken down by glycogen phosphorylase. And you could imagine that this structure has a lot of more branching than what I'm showing you here as well. Questions about this? Yes? Does a difference in bond energy come from steric hindrance? The answer is yes, partly. That's correct. Yep. Good question. Okay, well today, instead of having a song, I have something a little different for you. And it's something that I wrote that has never been put to music. So I'm always looking for people to put things to music and or to record my stuff. So if you're interested in either of those, let me know. Today I have something called an Ode to the Pentose Phosphate Pathway. And I'm going to read it to you. Okay. I need erythrose phosphate and don't know what to do. My cells are full of G6P and NADPH. But I just hit upon a plan as simple as can be. I'll run reactions through the path that's known as PPP. In just 2 oxidations, there's ribulose 5P, which morphs to other pentoses, each one attached to P. The next step, it is simple, deserving of some praise. The pentose carbons mix and match thanks to transketolase. Glyceraldehyde's a product, cetoheptulose is too, each with a trailing phosphate, but we are not quite through, even though many of you are leaving. Now 3 plus 7 is the same as adding 6 and 4. By swapping carbons back and forth, there's erythrose P and more. At last I've got the thing I need from carbons trading places. I'm happy that my cells are full of some transaldolases. Okay, see you guys on